everyone. We're going to go ahead and get started with our first speaker, Josh Davidson, on the topic of identifying the source of the Lower Cinnamonian mantle shell.
Yep. Go ahead and start. Okay. Thank you. So I'm going to be talking about identifying the source of the Lower Cinnamonian mantle shell of East Texas. And this is for my thesis, but I don't have all my data yet, so I'm just going to be talking about what I'm going to be doing, I guess. So, to jump right in, so the Lower Cinnamonian mantle shell is an argillaceous mudrock that occurs between the Butte limestone and the Woodbine sandstone in the East Texas field. This mudrock has been studied for several decades, but the sediment source of this rock is still somewhat misunderstood. So prior studies have indicated that the sediment comprising the mantle could have come from multiple sources, one of them being the Sabine uplift, which is going to be very important to my study and a part of my hypothesis. So a little geologic background on the mantle. As I said, it's a clay-rich mudrock that was deposited during the Lower Cinnamonian in East Texas and Gulf basins. It is the basal member of the Lower Eagleford, and it lies between the Butte limestone and the Woodbine sandstone. As far as deposition goes, this basin began to form during the late Triassic to early Jurassic due to the rifting and breaking apart of Pangea. And this was during the flooding of the North American continent, which established the basically established the Western Interior Seaway. Some of the tectonics and structural features that are that influence this basin and this mudrock are the Ouachita uplift up here to the north, the Sabine uplift to the east, which is a post-Jurassic large dome-like tectonic feature that became episodically active during the late Cinnamonian. You also have the Llano uplift over here in south central Texas and associated San Marcos Arc. You have the Sin depositional Luan salt features and the Sligo and Stewart City reef margins that built up here in the Albion and Birimian, and that was in the Texas Gulf Coast. So some of the previous studies that I'm going to mention here are what I'm basically going to be building my study off of. So you have the first one I'm going to talk about is Bailey et al., who actually identified the mantle in 1945. And I took this log from his paper, and I'm kind of using it to reference mine. And you can see the mantle interval is located right here between the Woodbine and the Butte. Another one is Barrett and Goodson, who tested reservoir quality of 9 Eagleford and Woodbine sandstone wells in Tyler County, Texas. And the methods in their study involved analyzing foram abundances and diversity changes, but I'm focusing on some of the logs that I took from their studies. And the mantle surface is right here in brown, and I'm using that to reference with mine as well. The most important study is Denham-Breyer 2016 A and B. And I guess the reason is because they really emphasized that the mantle shell was the first depositional episode signifying a shift from carbonate to siliciclastics of the Gulf Coast. And they were also the very first to generate an isopac map, which is displayed right here in the bottom right. This isopac map was basically correlated across the San Marcos Arc and into the East Texas Basin in South Texas and East Texas. And it illustrates a northeast to southwestern depositional trend, which indicates the direction of source, which is going to be important to my study because I'm going to be doing something similar. And I'm also going to be building off of their study and the study of 2 previous grad students that graduated over the past couple of years. So there have been problems in the geologic literature and in the history regarding the mantle because you've had different geologists publish papers calling the mantle different things. And that's because there's a sharp facies change when you move from northwest to southeast. And what I'm going to be doing is I'm going to be mapping from Dr. Din's study, and I'm also going to be mapping from the 2 previous studies of the grad students, Marissa English and Sam Patterson, to the south over here in this yellow polygon here. And the Sabine uplift is right here. So I'm going to be mapping to the south of the Sabine uplift. And what I'm going to be looking for is that facies change from fine grained siliciclastics to coarse grained sandstones. And if I see that, that will substantiate my hypothesis. And I guess I need to mention my hypothesis. My hypothesis is that the Sabine uplift is the sediment source of the Manus. So if I see that in well log, that can be an indicator that the Sabine uplift is the source for the Manus. And then I'm also going to go down to the BEG, the Bureau of Economic Geology, and I'm going to take a look at some cores, and hopefully I'll find some sandstones within those cores, or some coarser grained sediments within the Manus interval in those cores. And that will also give substantial evidence that the Sabine uplift is the source of the Manus. And that's it for me. So thank you. Any questions?
Feel free to send your questions via chat. We do have one for you, Josh, and it
Okay.
is, how many cores do you think you might have to look at?
So as far as I know, there's only 1 or 2 cores within 2 counties that show this at the BEG. There's one in Tyler County and there's one in Polk County. And these counties and these cores are located just to the south of the Sabine uplift. So if I go there and I look at these cores and I see that I'll probably look at like 2 or 3. And if I see that there are coarse grained sediments within the Manus, that's going to help. So yeah, that's it.
Okay, do we have any other questions for Josh? Okay, well, thank you so much, Josh. We appreciate your presentation.
Thank you. Thanks, guys.
Okay, we will now go ahead and transition over to Quinn. See if she's here yet.
Oh.
Here she comes.
And then I'll spotlight and that's what I forgot to do last time.
Great. Okay, our next presenter is Quinn Nguyen, and she's going to talk to us about the topic of the effects of neural manipulation on the nucleus of incumbents on reward loss.
Okay, I believe I have to mute, so please be prepared, Quinn. All right, you're unmuted.
Hi. Okay. Thank you for introducing me. I'm gonna start sharing my screen. Okay. So today I'm gonna talk about the role of nucleus accumbens in reward loss. So in our lab, we study the neural circuitry involved in reward downshift, or RD, which is an animal model of reward loss. So RD includes 2 stages, pre-shift and post-shift. Pre-shift lasts for 10 days, and during this time, it would give the rats a 32% sucrose solution, which is highly rewarding for them. And then during post-shift, which lasts for 4 days, we would downshift the reward value to a 2% or 4% sucrose solution. What we consistently observe is that during the first downshifted session, or session 11, the downshifted rats would consume significantly less sucrose, and this reflects the fact that the loss of the preferred reward was really frustrating to them. However, from session 12 on, we begin to see the rats recovering their consumption back to baseline. And so because of this progress, we further divide the post-shift phase into 2 more stages, suppression and recovery. So using this animal model of RD, research has explored many brain areas and pathways involved in reward loss. But within the scope of our study, we will only be looking at the nucleus accumbens, or NAC, within the context of RD. So the NAC is a subcortical area in the basal forebrain. And one study shows that during post-shift stage 1, NAC neurons actually release less dopamine in downshifted rats compared to unshifted controls. However, not much is known about the NAC during post-shift stage 2. And so our study aimed to explore the role of this area in the recovery phase of RD. And based on previous literature, we predicted that NAC excitation would increase sucrose consumption during stage 2 and increase the recovery rate. NAC inhibition would decrease the recovery rate. So we had 48 male Wilster rats in our study, and to manipulate the NAC, we used a chemogenetic technique known as designer receptors exclusively activated by designer drugs, or drugs for short. So this involved the intracranial infusion of a viral vector construct that carry an engineered receptor. And this receptor would later be activated by the injection of clozapine oxide, or CNO, which is the activated drug for drugs. And control animals in our study would receive a vehicle injection that does not have any effects. So we had 3 dread conditions: inhibitory, excitatory, and control, and 2 injection conditions, including CNO and vehicle. And so ultimately, we had 6 groups of animal in total. So after surgery and recovery from surgery, we moved into 14 days of RD, as I described earlier. And our prelim results show that NAC inhibition did not have any effects on RD. However, NAC excitation actually reduced the sucrose consumption and slowed down the rate of recovery, as you can see here on session 12 and 13. And so this is the opposite of what we predicted for NAC excitation. So one possible explanation for this finding is that we might have activated GABAergic neurons within the NAC, which might have led to less dopamine release and more behavioral suppression. Despite this, we were not able to verify this because of one limitation of the DREAD technique is that it is area specific and not neuron specific. Despite this limitation, our study was the first to explore the role of the NAC in reward loss using excitatory drugs. And as part of our future direction, we aim to explore the NAC in connection with the basolateral amygdala using more specific drugs. And that is the end of my presentation, and I'm taking questions.
Thank you, Quinn. If anyone has any questions, feel free to put them in the chat or to unmute yourself and ask.
You should be able to unmute now. So you have the free ability to do it. So
There you go. We have a question. It says the last graph showed a decrease in consumption from days 13 to 14 in one of the groups. Was that a meaningful decrease?
Okay, I'm going to share the screen again just to. So there was, okay, in one of the groups, you mean like, yeah, no, we did not, we actually did not find a significant difference in that group. I think you were talking about the yellow one, like control CNO group. And the difference between session 13 and 14 was not significant.
We have another question that asks if what would you like to do next in this study?
Yeah. So one about future direction is that we would be looking at the connection between the NAC and the basolateral amygdala, which has been shown to be involved in reward comparison. So animals who had this area lesions, they cannot tell the difference between a 32% sucrose and a 2% sucrose. So we believe that this area is involved in reward comparison, and after having information about the difference between the rewards, it will send that information to the NAC, which might be involved in the emotional response that leads to the suppression of behavior. And so that would be our next step.
Thank you, Quinn.
Yeah,
We will
thank
now
you.
have our next presenter, Michael Segala, on the effects of light availability, prey capture, and their interaction on pitcher plant morphology.
Awesome. Thank you. Let me share. Great. Sweet. So yes, as you stated, the effects of light availability, prey capture, interaction on pitcher plant morphology. So the pitcher plant, it's a species of carnivorous plant that grows pitcher-shaped leaves throughout the growing season. And carnivorous plants typically grow in these bog-like environments that are characteristic of having high light and water in their system. This allows these plants to grow leaves that capture prey for an influx of nutrients like nitrogen and phosphorus. This does have a trade-off, although, because as these prey capturing leaves have a reduced photosynthetic efficiency. Why can't I? Ooh, here we go. In this experiment, we set out to test how resource availability can affect pitcher plant morphology, so the size and shape of these pitchers that they produce. We've haphazardly selected 80 plants and randomly assigned them to these 4 treatments in a cross-classified design, testing the effects of light and prey availability. We had the 4 treatment groups: the control, which received full sunlight and were fed to simulate prey capture; an unfed group, which also received full sun, but were prey excluding, causing them to have reduced nutrient availability; a shaded treatment, which received prey, but had shade structures above them, which reduced their light availability; and an interaction treatment that had reduced light as well as prey excluded. Now, an important point about these plants is that the pitcher opening is correlated to prey capture. So a larger opening captures more prey and a smaller opening captures less. We hypothesize that those in the shade treatment will be light limited, so they'll exhibit a photosynthetic morphology in which they'll focus less on prey capture and try to maximize light absorption by producing pitchers that are taller with smaller diameters compared to the controls. The unfed treatment will have plenty of light, but will be nutrient limited due to the loss of prey capture and that influx of nitrogen and phosphorus is good for building block materials, and will exhibit plants with overall reduced growth rates, which should be measured by the number of pitchers produced and the sum of their heights. The interaction treatment plants will have reduced light availability as well as be prey excluded. So we hypothesize that they will take on this photosynthetic morphology of these taller, smaller diameter pitchers, but will also have reduced growth rate from the lessened nutrient intake. We ran our field experiment from spring to fall, and although we got tons of great data, here are just some of our main findings. So we have a bar graph here. On the X are our treatment groups, unshaded versus shaded, and unfed versus fed. And on the Y, we have average pitcher diameters. And as you can see by the graph in the attached ANOVA, there is a significant effect of shading on the diameter of the pitchers. So this supports our hypothesis that plants that are light limited will shift resources from carnivory and invest in light absorption and take on this photosynthetic morphology. And then this next graph here, again, the treatments on the X, unshaded versus shaded, unfed versus fed, and the average sum of pitcher heights, which is a way of measuring growth. And the graph with the ANOVA show a significant effect of prey exclusion on plant growth. So those that were prey excluded were nutrient limited and had reduced overall growth. Ultimately, these results support Givenish's cost-benefit model, which states that botanical carnivory is only advantageous under conditions of high light and water availability. And this study really exemplified that. As you can see in this first picture, this is from early on in the season, in which the plants are receiving plenty of sunlight and they're taking on this very typical plump, big opening pitcher morphology, ready to capture, eat a whole bunch of bugs. Then here in the second picture, this is from later on in the season, and this shows that as competing vegetation overtakes, so you got tall grasses and shrubs, the pitcher has become light limited. And although that looks like a blade of grass, that is in fact a pitcher plant. And the exciting part about this particular morphology is that Sarracenia lata is not widely documented to take on this form of practically no opening and appearing grass-like. So we were able to term it, Dr. Horner and I, as pseudophylodia. So that was pretty cool. So again, this shows that when pitchers become light limited, they will abandon carnivorous investment because it's no longer beneficial to the plant and take on this purely photosynthetic morphology. So in summary, our experiment supported our hypotheses that resource limitation can affect pitcher plant morphology. It supports Givenish's cost benefit model that botanical carnivory is only advantageous in conditions of high light and water. And lastly, kind of big picture, this shows support for frequent burns and proper conservation of these bog habitats, because without them, competing vegetation can grow unchecked and pitcher plants will begin to look like those in this top right picture. And after a couple of seasons, it's gonna have a really major impact on fitness and the survivability of the species in general. So thank you for listening. And I would like to give special acknowledgements to funding sources from the CERC and the Atkin funds, as well as shout out to my committee of Dr. Horner, Dr. Hale, and Dr. Williams. And a special shout out to my graduate student partner in crime, Paris, who helped with field work throughout the summer. And with that, I will field any questions.
Thank you. So we do have a question. Are you getting this published?
Yes, it is. Me and Dr. Horner are currently working on trying to get it published.
Okay.
Hopefully
Where were
this
these
summer.
grown? Where were
Well,
these grown?
So yes, this was grown in a private landowner's bog in Leon County, Texas. So what was it, like 2-3 hours away from Fort Worth in the direction of like Louisiana?
I guess just as a follow-up, is that where they're native?
Yeah, so they're native all throughout the southeastern United States. And then sort of, they were much, I mean, if you look back like a couple hundred years, they were much more spread out, much more common, but due to just kind of land use changes, kind of this fire mitigation by humans and whatnot, their populations have really shrunk and become very fragmented. Yeah, they're native to Texas and Louisiana.
Great. Thank you, Michael. We appreciate your presentation.
Awesome.
Good luck with the publishing.
Thank you so much.
Okay, our next presenter is here. So our next presenter is Shelby Miller, and she's going to be presenting on the effects of chronic unpredictable stress on cognition and AD-like pathology in mice.
Hi, thank you, Sophia. So real quick, I just wanted to thank you all for coming out to listen to us present and take time out of your day to do this. My name, as Sophia said, is Shelby K. Miller. I'm an undergraduate research assistant who works in the neurobiology of aging collaborative that is run in joint by Dr. Michael Chumley and Dr. Gary Boehm. Along with myself, my two other presenters are my fellow lab mates named Connie Leonardos and Evan Shanley. Each of us have broken up our presentation to discuss different aspects of it with you. But while doing so, because we have so much information that we found relevant, whenever I present our poster, I will, just to give you a little bit of a heads up in how we're doing this, I will zoom in on different portions just so you all can better easily access the data and figures that we show. But I will go ahead and share my screen now. So we'll start it off with Evan giving you a little bit of an introduction and background on Alzheimer's disease, which is largely what we research in our lab, along with other neurodegenerative diseases. I will run through the methods with you. Connie will go through the results, and then we'll finish and close it off with Evan pointing you towards future directions that we are considering for our lab. So Evan, if you want to go ahead and take it away and give a little introduction.
Yeah, thanks, Shelby. So the paper that we're going to be talking about today, or the study rather, was primarily aimed at discerning the relationship that might exist between the application of a chronic stress paradigm and the development of an Alzheimer's disease-like phenotype that we were able to induce previously in our lab using administrations of LPS, which is essentially just a pro-inflammatory bacterial mimetic. It activates the immune system. And so that exacerbates sort of the development of some of those neurodegeneration symptoms in the C57BL6 mice. Given how pervasive both Alzheimer's disease as well as chronic stress are within the United States, affecting 5.8 million people and 2/3 of the population respectively, we thought this question would be really important and sort of primed for further investigation, leading to the hypothesis that the chronic unpredictable stress would be able to exacerbate the LPS-induced Alzheimer's disease-like pathology within this strain of mice. Now, Connie's going to discuss the methods for you guys.
Or I'll actually take over the methods, Evan. So let me go ahead and scroll down to the methods for you all. So before I directly jump into the methods, I wanted to discuss a little pilot study that our research lab had previously done that we used as sort of a model to then jump into the experiment that I will introduce to you all in a second. But a pilot data that we produced showed that 7 days of unpredictable stress is associated with impaired cognitive function in contextual fear conditioning. So this is looking at unpredictable stress, but we kind of wanted to take this 7-day paradigm and move it into looking at chronic unpredictable stress. So as Evan said, the strain of mouse that we used in the actual experiment that I'll now introduce you to is the C57BL6 mouse. All of them are male, and we divided them up into 2 groups. So we had the chronic unpredictable stress group, which I will refer to as CUS, and then we had the group housed controls. So those mice who were in the group housed controls, we kept in standard laboratory conditions, not in isolation. And then for those who were in the CUS group, we exposed them to 6 different stressors, which are highlighted in this blue box up here. So throughout 21 consecutive days, they experienced these stressors every day. This included having a tilted cage, making their bed that they sat in wet, putting them under restraint stress, making them do a forced swim for 5 minutes, having an empty cage so there was no socialization and experienced isolation. And then we would take away their nestlets so that they weren't provided a nestlet to sleep in during the night. All of these stressors we presented the mice to in a very sporadic order every single day. And this was to control for not having any of our mice habituate to the stress. We really wanted to hone it down on a model that was really accurate of chronic unpredictable stress. So towards the end of those 21 days, starting when there were 7 days left in the study, is whenever we injected our mice with either LPS or saline. So taking the CUS group and the group housed controls, we divided those in 2, and they were either administered saline as a control to control for the stress that the mice experience when being injected, or they were administered LPS, and this is to have them develop a pathology of a late set of Alzheimer's disease. And then following the end of those 21 consecutive days and then 7 days of injection toward the end of the study is whenever all of our mice underwent contextual fear conditioning to examine learning and memory. So it's the natural behavior of a mouse to freeze upon being administered a shock. And so when we place them in an environment where they received the shock the very next day, if they exhibit learning and memory, they should display freezing behavior before they are even administered the shock. And then following contextual fear conditioning, hippocampal tissue was collected from all of our mice to quantify what's called amyloid beta. This is a protein which aggregates to form plaques that disrupt neuronal communication in Alzheimer's disease. And although we don't know the direct cause currently, amyloid beta is a major marker of late-set Alzheimer's disease, which is why we look at it so heavily in our mouse models. So now I will shuffle it over to Connie for her to discuss the results with you.
Great. So Shelby's going to pull up this pilot data up top. If you can see on that first graph, And this is from those 7 days that we did of unpredictable stress compared to a group that was just in isolation, as well as our group control housing. So as you can see from the 7-day study, we determined that mice in the unpredictable stress group froze less compared to both isolation and control groups, which indicates that there is greater cognitive dysfunction in the unpredictable stress group. Because of this, we were able to use that data to switch over into our 21 days of chronic unpredictable stress. And in this study, we also found that there is a significant main effect between CUS and the control when it comes to percent freezing during CFC. And then in the third graph, you can also see that there is a main effect that was approaching significance such that the CUS animals had a higher A beta protein when compared to the controls. While this wasn't quite significant, it was trending in the hypothesized direction. Because of this, we were able to conclude that 21 days of CUS was associated with Alzheimer's-like pathology in C57 black 6 male mice. And I'll pass it back to Evan for future directions.
Yeah, so in terms of future directions, there were 2 primary modifications that we thought might be beneficial to make to the current experimental procedure. The first of which would be using a slightly stronger variety of LPS in order to further investigate the relationship between stress and inflammation. And the second being the addition of a second behavioral test in order to further sort of operationalize the potential cognitive deficits that these mice could be experiencing and whether or not stress is impacting that to the same degree that it could be impacting some of the histological markers. From a more broad point of view, however, we think that understanding the mechanistic relationship between something like a chronic unpredictable stress paradigm and Alzheimer's disease pathology could be super beneficial given that not a lot of treatment options are available, and with how many adults are suffering from both stress as well as Alzheimer's, this could really pave the way for a lot of great future research if a more mechanistic target could be identified. That's pretty much it. Are there any questions?
Thank you. Please feel free to put your questions in the chat or to unmute yourself. Okay, well, we think you probably answered the question on future research since you did tell us about that, and it is time to move to our next presenter. So I want to thank you all so much for your presentation.
Thank you.
Thank you.
Okay, our next presenter is Malia Iragwan, and her presentation is on the role of multi-scripturalism on novel orthography learning.
All right, I will go ahead and share my screen. My name is Malia Iragwan, and I conducted my honors thesis research in the Dr. Santani's lab, the genetics of auditory perception and plasticity here at TCU. So I will go ahead and zoom into this introduction. Since 1980, the use of a language other than English at home has increased by 148% in the United States. As the United States becomes more lingually diverse in the coming years, more people will become multi-scriptural or fluent in multiple print systems. There are many benefits to acquiring additional orthographies, but orthography learning becomes more difficult after the reading acquisition period closes at 18 to 19 years old. Bilinguals have shown an advantage in language acquisition in adulthood, but no research has studied the effects of multi-scripturalism on reading acquisition. Given the connections between the reading and the language networks, we investigated if the bilingual language advantage would also carry over to orthography learning. We hypothesized that individuals who are familiar with multiple scripts will have an advantage in novel orthography learning compared to those who only know one script. 70 young adult participants completed a background survey and assessment session over Zoom to determine their eligibility. Of the 70 participants, only 26 were eligible, and they completed a training session to learn Hebrew vowels and consonants. Directly after finishing the training session, they completed a post-knowledge learning check, which included our dependent measures of letter ID, automaticity, and decoding. Lastly, participants completed the same dependent measures 7 days later to examine effects after a period without training. Only 14 participants in this point in time had completed the full study. So in our current results, Monoscriptural participants showed a decrease in Hebrew performance on the letter ID task, but an increase in performance on the RAND task. And there was no significant difference between mono or multi-scriptural groups on the decoding task. There was no effect of time or group. So our current results show and suggest that there may be no difference or no multi-scriptural advantage in novel orthography learning, which may be explained by the grain size hypothesis. The grain size hypothesis suggests that the ability to pick up a new orthography depends on the match of the grain size between the orthography that you know and the orthography that you are trying to learn. So grain size is the smallest grapheme in a language or in an orthography, and they fall on a spectrum from shallow to deep. So shallow orthographies include diacritic Hebrew, like the one used in this study, and they have short and simple graphemes, while deep orthographies are more complex, like the English language. The theory posits that one would be able to pick up an orthography easier if the grain size they know matches the one they're trying to learn. So our study only included English, Spanish, and German participants. So in future research, we hope to explore more participants with a wider range of orthographies to see if the grain size may affect their ability to pick up a new orthography. So if our current results hold true, we do plan to collect 40 participants before finalizing a data set. But if these do hold true, then this suggests that there's no multi-scriptural learning advantage. This may be this shows that the language and reading acquisition mechanisms may be completely different. So for example, humans are wired for language acquisition and may be able to become fluent in multiple languages. However, this does not mean that they may be able to learn how to read in all of those languages. Evolutionarily speaking, reading acquisition is relatively new, and this research supports that the mechanism to learn reading is different than the one to acquire language. These results lead us to question if there is a limit to how many orthographies that we can store if the reading acquisition period affects their ability to learn a new language. Maybe on children, this advantage does show in multi-scripturalism, or if the orthography fluency affects how they're going to be able to pick up a new one. We will continue data collection and evaluate the effects of multi-scripturalism, orthography depth, and fluency on acquiring new alphabets. I would like to give thanks to Dr. Santani, Dr. Fikar, and the research assistants for all your assistance. Thank you for listening, and please ask questions in the chat.
Thank you, Malia. That was very interesting. If anyone has questions, please feel free to post them. So I'll ask in the meantime, when do you anticipate having all of your participants' data completed?
We are currently remodeling our data and how we're going to move forward from here, because we noticed that a lot of the participants on the training and the post-knowledge task were reading incorrectly. So they're supposed to read from right to left, but a lot of participants were not paying attention and actually completed some of our dependent measures incorrectly. So we plan to host those sessions over Zoom to make sure that all participants are following correct instructions. And we are considering starting this over the summer. And if not, we are definitely going to continue it into the fall to reach that 40 participant mark that we are aiming for.
Great. What other languages would you hope to study in the future with this same model?
Since we only have English, German, and Spanish so far, we're hoping to get a wider range of orthographies, maybe some people with more shallow orthographies. It kind of depends on our participant pool, but we hope to maybe get some Japanese or Arabic, because Arabic also is read from right to left, so that may influence their ability to pick up the new one reading from right to left.
Okay, I don't see any more questions in the chat. Does anyone else have any final things to ask? All right, Malia, well, thank you so much. We appreciate your presentation. Okay, looks like our next presenter is here. So we'll go ahead and introduce Carice Kang, and The topic is effects of pollen limitation on seed production in the pale pitcher plant.
Let's see, is my screen, let's see, sharing properly?
Yep, you're good.
Awesome, thank you so much for the introduction. All right, so pollination is an essential process that maintains ecosystems from one generation to the next. Pollen grains are produced on the anthers, which are the male organ of the flower, and they are transferred to the stigma, which is the receptive female organ of the flower. And this is done by a pollination vector, such as an insect, an animal, or something like the wind. So once that pollen grain is on the stigma, it produces a pollen tube that reaches down the style all the way to the ovary. The male gamete travels down this tube to the ovary where it fertilizes the ovule, or the female gamete of the flower. And through this process, seeds are produced, and we're able to continue plant populations from one generation to the next. So under this system, more pollen grains are produced than ovules. Ovules are the resource expensive female gamete, and the less resource expensive pollen grains compete with each other for the opportunity to fertilize these ovules. However, this system can be disrupted if not enough pollen grains land on the stigma. Not every ovule has the opportunity to be fertilized, and the flower produces fewer seeds than it has the full capacity to produce. And this is known as pollen limitation. Now, pollen limitation has been observed in different species across different plant families. However, some groups of plants have not been observed as extensively as others, and carnivorous plants is one of these groups. In fact, there were no pollen limitation studies done in any carnivorous pitcher plant from the Gulf Coast of the United States, where the majority of these species are. So with all of this in mind, our study looked at a population of pale pitcher plants, or Sarracenia alata, in Central Texas. And we asked three main research questions: whether floral herbivory could affect pollen availability, whether decreased pollen receipt could impact offspring quantity or the number of seeds that are produced, and then finally, whether pollen limitation or decreased pollen receipt could affect seed quality. So in order to address our first question, we looked at flowers that had been damaged by floral herbivores and flowers that had been protected from floral herbivory. And we measured both the number of anthers found in each flower and the anther mass. And as you can see in our results, flowers that have been damaged by floral herbivores have significantly fewer number of anthers and also mass of anthers compared to undamaged flowers. So floral herbivory can significantly decrease pollen abundance in damaged flowers versus undamaged flowers. In order to address our second research question, we took flowers from throughout the bog and we divided them into three treatment groups. A low pollen group, which only had access to pollinators for part of the flowering season, a control group, which just had natural pollination for the entire flowering season, and a high pollen group, which in addition with natural pollination was also given hand pollination from donor flowers randomly selected throughout the bog. So in the fall, we collected seeds from these three treatment groups, and we measured both the number of seeds each ovary produced and also the total mass of seeds per ovary. And as you can see in our results, there was pollen limitation in the year 2019 when we did our study. Our control group much more closely resembles our low pollen group than our high pollen group. So our high pollen treatment had produced significantly more seeds than either our low pollen or our control pollen group. So now that we know that pollen limitation can occur in the species, we looked at whether pollen limitation could affect not only the number of seeds produced, but also the quality of the seeds. So we did this by measuring the mass of an individual seed and also by putting these seeds through germination treatments. So in that first graph below our third research question, you can see that the average individual seed mass was not terribly different between our groups. So the average seed in our low pollen group was about the same mass as the average seed from our high pollen group. When we germinated these seeds and measured the proportion of seeds germinated, the average germination time, the germination synchronicity, or just how closely together these seeds germinated, and also the germination rate, we did not see a significant difference between any of these treatment groups. So an overview, floral herbivory can significantly decrease pollen abundance in damaged flowers versus undamaged flowers. However, in the year of our study, 2019, in the overall population of flowers, there was a low proportion of flowers that had actually been damaged, less than 2%. However, pollen limitations still occurred during this year. So this tells us that while floral herbivory may contribute towards pollen limitation in some years, during the year of our study, there's probably other factors driving pollen limitation. That being said, pollen limitation did not seem to have any discernible effect on seed quality as measured by individual seed mass or the germination benchmarks that we looked at. So in conclusion, pollen limitation occurs in Sarracenia alata, and it may pose a conservation risk when paired with other ecological disturbances. I'd like to thank Dr. Hale and Dr. Williams for being on my committee, fellow lab members for all the help they had in the field, and of course, RCAF, CERC, and Atkins funds for helping making this research possible. Thank you.
Thank you, Carice. We have a question for you. How many seeds or plants were in your study?
Let's see. So for our first research question, sorry, let me just bring this back up. Let's see, for our first research question looking at damaged versus undamaged flowers, we had 26 flowers in our damaged group and 30 in our undamaged group. And again, that's just because there's such a low proportion of flowers that had been damaged that year. It was actually kind of hard to find them. For our pollen limitation experiment, the second one, we started off with 30 flowers per group. However, due to flower loss kind of over the season, like wild hogs sometimes went through the bog, sometimes we couldn't find our tagged flowers again. The number of flowers that were recovered for each group varied drastically. Actually, our high pollen group suffered the most. So we actually only managed to collect 6 flowers from that group. So that is one of the reasons we probably want to rerun this experiment during a different field season, because there's such a loss of flowers over our research period. And then of course that has affected our sample size for the last group, which depended upon the second experiment.
And with a few more seconds left, if you could answer, do certain pollens dominate the air? And do you think that that affects those with less quantity of pollen in the environment?
Let's see, to clarify this question, is this about pollen clogging from other species which are not pitcher plants?
I don't know that I can clarify, so yes.
I'm gonna go off of that assumption then. Because these flowers are shaped kind of like a closed capsule, the receptive stigmatic surface is actually kind of tucked inside the petals, and the only way pollen can get to it are through bumblebees, which are kind of the specialists. Well, they are the only known pollinators for this species. So for this particular group of plants, I'd be less concerned about pollen clogging from other species, because they really can't get to those stigmas. They can't just blow in randomly. They need to be transported by bumblebee.
Thank you so much. Thank you, Carice. We appreciate your presentation. Our next presenter, our next presenter is Christopher Wilson on the analysis of river flooding impacts on the Trinity River in Tarrant and Dallas counties.
Hi. Now, not sure I can really live up to the bumblebees, but I will do my best. Let me make sure that it's sharing the right screen. I don't think that one is the right one. Here we go. Right. So I worked on this project with Megan Alexander, who is another undergraduate student in the Department of Environmental Sciences, and our faculty advisor was Dr. Gebra Michel. And so for this project, we were looking at really one of the biggest of the big problems going forward in urban areas, and that's major flooding especially flooding as it affects property damage and human life. And so for this project, we used a combination of USGS flood gauges, FEMA floodplain mapping, as well as the elevation maps of Tarrant County to look at the Trinity River that flows through the middle of Fort Worth to look at kind of the possible impacts of current flooding situations and possible flooding situations that might not have happened yet, but are probable. And this is really important because flooding annually causes somewhere around 18 to 20 billion dollars in property damage and kills hundreds of people. And you know, we're not exempt from that here in Fort Worth. And so our three main objectives in our project were to determine the potential risk to human life from these flood stage levels, economic impacts, and then risks to critical infrastructure. And so I'll hand it over to Megan to talk about our data.
Yeah, so for our data, it's also important to acknowledge, I think before it was mentioned that our study area included Dallas County, and it actually only includes Tarrant County. So if you could zoom into the first graph in our data that was created in ArcGIS Pro, it shows population density in Tarrant County and how it follows the flow of the Trinity River and subsequent floodplain locations. And so, as you see here, we have our flood gauges at flood stages 0, 15, and 28 feet. And so overall, the locations of stream flow are in slightly higher populated areas as compared to the lighter orange color in the corners, which are the lower populated areas. And so for our second data graph that was created, it shows these flood gauges at their levels, and the flood wave propagation data taken from these gauges historically, as well as elevation and discharge data, were used to create a layer projection showing an accurate 100-year floodplain event. And so these lighter blue colors that you see in the graph itself, those are the floodplain locations. Where the bright red color is, that would be buildings or infrastructure that we found that occur within the floodplain location. And then the lighter gray/purple areas, those are buildings that are unaffected. And so as you can see in Tarrant County, there are quite a few buildings that are impacted by such a flooding event. And so as you can see at the bottom as well, the third graph, that shows our elevation data. And so that's just a reference to natural hydrology and the flow of our riverine system. And so as far as the methodology, we used a multi-step process to analyze publicly available GIS data falling within this floodplain. And so first we found population, property, and infrastructure data for Tarrant County. And then we created our maps, and then we used statistical analysis as well as processing tools available in ArcGIS to determine the impacts, not only just in a spatial way, but also monetarily. And so our results found that like overall damage from a 6-inch flood in all affected properties within the floodplain projection would amount to almost $1 billion, so 770 million in Tarrant County alone. And it's also important to acknowledge that using our geoprocessing tools, we clipped the buildings into our floodplain area to find that 3,430,503 square meters were affected property-wise. And so while there is a significant population at risk, it's important to note that this is 100-year floodplain data. And so it's not very common that flooding at this level would happen on a normal or regular basis. And so at the bottom, you can see a graph showing
as flooding increases in inches, the amount of money taken to kind of alleviate those efforts would
Right.
increase as well.
Great. So now I suppose we'll take questions.
Thank you all. Does anyone have any questions they want to share in the chat or unmute and ask?
Well, if there's no questions, I will mention one thing real quick, that we were very limited on our infrastructure analysis by the lack of publicly available information. And so if we were to expand on this study in the future, we would likely try to work more with the city of Fort Worth to get access to some of that data that is not publicly available for security reasons.
Great idea. Okay, well, thank you so much. We appreciate y'all's presentation. Good luck on your future research.
Great, thank you very much.
Thank you.
Okay, it looks like our next presenter is here. So I'll go ahead and introduce Amy Lamb, who is presenting on mapping fertilizer runoff in the Mississippi River Delta.
I'm also presenting with Graham Rice, who is like right next to me.
Yeah.
So does he have permission to share screen?
Sorry, my bad.
I think they'll be able to give him, is he going to share his screen?
Should be able to just hit the share screen. There we go.
I'm muted. Are you muted?
Mhm.
All right. Good afternoon, guys.
Graham, we cannot hear you right now.
Mute myself. Thanks. So we're doing our presentation on the nitrogen and phosphorus runoff within the Mississippi River Delta, and I'll let Amy take it away for the introduction.
So for our study area, it's focused on the Mississippi River Basin.
Amy, if you could unmute yourself.
Sorry, our study area focuses on the Mississippi River Delta or Basin, which is the 7th largest river basin on Earth. It has an area of over 2 million hectares, which is equivalent to 4 million football fields. So it's really big. In the river basin, there's the Mississippi River, the Gulf of Mexico, and 40% of the wetlands in the continental United States, mostly in Louisiana. Previous studies have shown that fertilizer runoff from agricultural lands has led to excessive levels of nitrogen and phosphorus, which are inorganic plant and algal nutrients in the river basin. This has led to water pollution and eutrophication. Our research will focus on mapping the levels of the nitrogen and phosphorus levels across the river basin and along with the land covers of the river basin itself. To be more specific, the objectives of our research is to analyze the land cover of the Mississippi River Basin, map the nitrogen and phosphorus levels found throughout the basin, and to find the correlation between the two of them.
Thank you, Amy. So our inspiration for this project was actually inspired by a rivers and landscape discussion on the Mississippi River. During one of the lectures, I saw a kind of a photo of how the basin itself kind of all filters down to the river delta, and I kind of made me think of how all of our farmland is in that exact area and how these fertilizers and other chemicals end up in the Mississippi River Delta. So to start, we acquired our data from the ArcGIS All portal. We found the Mississippi River Basin feature and the Mississippi River system, including all the rivers, all the tributaries that lead into the Mississippi River and then go out to the Delta. Our land coverage data, seen in the photo below, was acquired from the National Land Cover Database, which is offered by USGS. And using that, we were able to mask the roster and match the boundaries of river basin, and kind of filter out all the parts that weren't a part of the basin itself. We then used research data from the 2019 USGS Sparrow model, and through that, we were able to identify levels of nitrogen and phosphorus among different catchments in the basin, which can be seen in the results and discussion. And we were able to georeference both Sparrow models to match the Mississippi River Delta from the phosphorus and the nitrogen. And so if you look at the figure 3 and figure 4 under results and discussion, you can see, I'll zoom in a little bit for you guys. You can kind of see these higher levels right around the Mississippi River itself, which is like also a high concentration of farmland, as seen in the National Land Cover Database. And these, as it filters down through the Mississippi River, it just all kind of grows, and the amount of nitrogen levels grow, and going into the delta itself. And it's kind of the same trend with phosphorus as well. You can see the darkest levels are right around the Mississippi River Delta. And so we can kind of attribute that to the fact that there are a lot of, try to zoom into the land cover percentage as well. There are a lot of farms and agricultural areas right around the river basin, which like doesn't really make much sense because the soil is very fertile, but the processes using to grow these agriculture can actually have very bad effects with runoff into the river delta. These fertilizers, they really implement the usage of nitrogen and phosphorus, and this is seen in like a really detrimental thing going into the Gulf of Mexico, which can lead to eutrophication and hypoxic zones. In terms of acting against this, we didn't do very much further research into that, because in terms of that's kind of our crop land right now. That's the kind of what they call the heart of the United States for a lot of agriculture. And so there's definitely some consequences of that, but in terms of actually taking action, we didn't present any former ideas. Thank you. I'll now be taking questions.
Thank you. And everyone, please feel free to put your questions in the chat. We do have a question for you all. How might land usage as it pertains to nitrogen and phosphorus loads affect the spread of your data in future studies?
Could you repeat that question one more time?
Yes, it's how might land use as it pertains to nitrogen and phosphorus loads affect the spread of your data in future studies?
So I think a lot of the spread for the data, the reason why the runoff is so high in these areas is because so much water is being pumped in these areas, and that's increasing the runoff load. Whereas in a system like a prairie, you're not going to have as much irrigation on the system. So I think as farmlands are continuing to change and continue to move, I think you may see less of that runoff from irrigation going into the river. But it does make sense that a lot of the agricultural land is on a basin that's has very fertile soil. So it'll be definitely interesting to see how that develops over time.
Okay, well, thank you, Amy and Graham, for your presentation. Very informative.
Thank you.
Thank you.
Okay, our next presenter is Lauren Edwards, and her topic is environment sensitive probes based on scoric acid scaffolds.
Hello, thank you. Okay. Can you see my screen?
Yep, you're good to go.
Yes, so thank you for the introduction. My project is based on environment sensitive probes. So environment sensitive probes are molecules that have a set of photophysical properties that change in response to a change in their environment. The general mechanism by which these photophysical properties are measured is through the excitation of the molecule to an excited state and consequent relaxation to the ground state. And from this, we can get photophysical properties such as absorption, emission, and the lifetime of the molecule. And so what we're looking for, again, just to reiterate, is that to have one set of photophysical properties of the molecule by itself in a standard media, and then a shift when the molecule experiences an environmental change. And there are several mechanisms through which this can occur. I'm not going to talk about all of them. The one I want to focus on is conformational changes, since this is what our group is interested in. And this is when there is a change in the molecule from a twisted to a planar conformation, which allows for the molecule to be an environment sensitive probe, since this form has one set of properties that are different than that as a result of an internal rotation. And these molecules are aptly named molecular rotors and have several applications, but our group focuses on measuring viscosity and polarity of the media. So the goal became to generate a multiplexing probe, or a probe capable of responding to several types of changes in the media. And an accessible route was using scoring dyes, which are based on the scoric acid scaffold shown here. And these are rather attractive because they have a lot of applications you can see. And they can be functionalized in a number of manners using several types of compounds, which would be denoted here as X and Y. And so with this in mind, we used 8-aminoclinolene as our functionalizing entity, since the nitrogen here can be manipulated by alkylation to further tune photophysical properties. So here are the reaction conditions we used to generate these 4 compounds of interest. And part of our goal was to have a simple synthesis from readily available starting materials to produce compounds in moderate to good yields, which we achieved. So with the mono-substituted hydroxyscoric acid, the absorbance was measured in solvents of various polarities, and we were looking for a linear correlation between the peak absorbance of the molecule in response to the polarity of the solvent. However, based on the low R-squared value, you can see that there was really no correlation between the absorbance spectra and the polarity of the solvent with this particular derivative, although the absorbance spectra was changing. So using the ethoxy version of the mono-substituted scoric acid under the same parameters yielded a better correlation. However, it was still not very good. But as we increased the complexity of the scaffold, we got an even better correlation with the 1,2 disubstituted scoric acid. But the real winner was the 1,3 disubstituted scoric acid, which had the best linear correlation. And this really goes to show that the gradual adjustment of the substituents and their placement on the same scoric acid scaffold leads to the design of a polarity sensitive probe while still maintaining the simplicity of synthesis. So the mono-substituted scoric acid derivatives for our purposes are non-emissive. So from here, we're really only going to be looking at the di-substituted derivatives. So when we plotted the emission as opposed to absorption of the derivatives as a function of polarity, we didn't find any correlation. But we took this one step further and tested how the emission of the 1,3 disubstituted scoric acid would respond to changes in viscosity of the solvent, and found that this derivative is responsive to changes in viscosity. And this was a really good sign because our goal was to synthesize a multiplexing probe, and finding that the 1,3 disubstituted scoric acid had only absorbance respond to changes in polarity and only emission respond to viscosity, we can have a probe that can measure both of these properties without one affecting the other. So it's been shown that for probes to be viscometers or rotors, the plot of some photophysical property as a function of viscosity must yield a slope greater than 0.1. So we did a more controlled experiment, which was using a mixture of various ratios of methanol and glycerol to tune the viscosity of our solvent. And as we increased the amount of glycerol and therefore the viscosity of the solvent, we saw a nice correlation and, more importantly, a slope that was greater than 0.1, which means that this 1,3 disubstituted scoric acid is a viscometer with the 8-aminoclinolines rotating around the scoric acid scaffold. We also looked at emission lifetime, which again is just a photophysical parameter telling us how much time the molecule spends in the excited state. And we plotted this as a function of viscosity and still got a slope greater than 0.1. And the point of doing this was just to ensure that this really is a viscometer and to remove any question of how changing the concentration or the ratio of methanol to glycerol might affect emission. So this was just a confirmation that we do have a rotor. So the conclusions that we came to in this project are that molecular rotors can be used to assess properties of the media, such as polarity and viscosity, and that the 1,3-disubstituted scoric acid with the 8 aminoquinolines is a multiplexing probe that measures viscosity and polarity. And we also achieved our goal of a facile and concise synthesis. I'd like to thank my group members as well as the members in the physics department for the development of this project. And at this time, I will be taking questions.
Okay, thank you, Lauren. Do we have any questions for Lauren? Lauren, do you have any future ideas for your research?
Yeah, so we've actually already taken this several steps further and created a library of compounds based on different isomers of the aminoquinolines. So I believe it's the 2, 3, 5, 6 aminoquinolines. And we're currently awaiting to see those properties to see if they are potentially even better rotors.
Okay, feel free to put your questions in the chat or unmute yourself and ask. Okay, well, I don't see any further questions at this time. So Lauren, thank you so much. Okay, we have our next speaker here, who is Marinata Dedet, and the presentation topic is on development of biocompatible graphene quantum dots with near-infrared fluorescence.
Hi, thank you. I'm going to share my screen. Can you guys see my screen? Okay, good. Hi, my name is Matt Dedet. I am a physics major. And today I'm going to present about the development of biocompatible graphene quantum dots with near infrared fluorescence. Oops, sorry. What if we could develop new methods of treating cancer? What if we could detect cancer cells without the need of biopsy and dyes? What if we could develop or eliminate the need for chemotherapy? Current imaging techniques being used for cancer therapy are limited, as they cannot detect a small number of cancer cells in the tissue, and some of them can be harmful. While the use of quantum dots have been poised to become an improved methods of imaging, there are only a few types actually that are known to emit light in the near infrared. So our lab has therefore had the objective to seek and develop these unknown types of quantum dots. So quantum dots are semiconductor nanoparticles that glow when exposed to light. In medicine, their physical, biological, and optical properties allow them to be used as imaging and drug delivery agents for cancer. They can easily enter and exit the blood. They can be tracked due to their fluorescence, and they're non-harmful to living organisms. In this research, we developed biocompatible graphene quantum dots capable of emitting light in the near infrared region. Near infrared light is better, for example, than visible light because it can penetrate the skin and blood deeper and still causes no damage to tissue. We've therefore used bottom-up and top-down approaches to synthesize these quantum dots and measure them for absorption and fluorescence. So the top-down approach involves breaking down a bulk material into smaller sized particles. You can think of it as slicing bread to have a toast, to have toast bread. And the bottom-up synthesis involves stacking up small particles into structures. So it's kind of like playing Tetris or building Lego structures. And from our bottom-up approach, we used a one-step hydrothermal reaction with a microwaving oven to derive GQDs from a glucose and liquid ammonia mixture and from L-glutamic acid. From our top-down approach, we used a UV-led photolytic reaction to derive GQDs from reduced graphene oxide. Okay, I'm now going to describe the top-down synthesis for RGQDs, which are reduced graphene oxide quantum dots. A mixture of RGO flakes, water, and sodium hypochlorite was used to was subjected to a UV light synthesis to obtain RGQDs. Using an 808 nanometer laser excitation, the resulting fluorescence spectrum was obtained on the left, and furthermore, absorption spectra was measured from the solution on the right. Due to the near-infrared fluorescence, these RGQDs are good candidates for our future cell studies. I'm also now going to describe the bottom-up synthesis of glucose and liquid ammonia-based GQDs. In this polymerization and nucleation of glucose into GQDs, a mixture of glucose and liquid ammonia was diluted in water and microwaved to give this gold solution. We also used 808 nanometer laser excitation to measure fluorescence, and we measured absorbance as well. The observed near-infrared fluorescence also makes these quantum dots good candidates for future cell studies. Now, for this bottom-up synthesis of L-glutamic acid, a sample of L-glutamic acid was melted at 210 degrees Celsius until it turned brown. It was then mixed with water and stirred for 30 seconds until the solution cooled down. Using 808 nanometer laser excitation as well, fluorescence was measured and absorbance spectrum was also measured. And we can also tell here that due to the near-infrared fluorescence, that these quantum dots are good candidates for cell studies. In conclusion, from this research, we can conclude that there are more near-infrared emitting types of graphene quantum dots in addition to the already known ones. We will further study these three structures for biocompatibility and cell studies. Another observation that we made is that there aren't many graphene quantum dots that can emit in the near-infrared region, as we've seen with multiple failed attempts at measurement. But being able to develop these three near-infrared emitting quantum dots is an amazing achievement in its own, because it brings us closer to simplify cancer therapeutics and save more lives. Does anyone have any questions?
Thank you so much. That's some really exciting stuff that you're doing there. So we appreciate your work and sharing it with us. Does anyone have any questions? We have a question that says, can you give details on how a light emitting property makes them good for detecting cancer?
How a light emitting property makes them good for detecting cancer? So basically you can, so due to their fluorescence, which is that light property, I'm guessing that's been asked or spoken of, due to their fluorescence, you can shine a light on them and they'll glow. And since, since near-infrared light is so deep that it can penetrate the skin and blood, you can see it. You can basically map out the position. And if it happens to be on the cancerous cell, for example, you can see the exact position as to where the cancerous cell is.
Okay, we have another question. Did the method of synthesizing them, I guess the quantum dots, affect their detection?
Yes. So what are called precursors are what glucose and, for example, liquid ammonia are. So the precursors themselves do not fluoresce. And so by synthesizing them, by making compounds out of them, you can then basically, depending on the synthesis that you do, the more fluorescence they can have. So yes, you can definitely do that.
Great. So I think that's probably all we have time, all the time we have for questions. So thank you so much. We appreciate you sharing your research with us.
Thank you.
Okay, our last presenter for today is Veronica Lyle, and her topic is pH-based cancer detection by graphing quantum dots.
Good afternoon. As previously stated, I am working with Dr. Namov along with Matt Daddett, who just presented, in pH-based cancer detection by graphing quantum dots. So an undeniable statistic is that the earlier cancer is detected, the greater the chance of survival. So my research has been investigating a potential method to increase our ability to detect cancer accurately. So a somewhat obscure yet frequent characteristic of many types of cancer is their pH difference from healthy cells, both internally and externally, due to metabolic changes that occur in the act of becoming cancer. So most notable is the significantly higher acidity of the fluid outside of cancerous cells relative to healthy cells. So as you can see, I've marked the internal and external acidity of healthy cells, of cancerous cells in red, and healthy cells in blue. So as already covered by Matt, our lab has been studying the use of quantum dots for bioimaging purposes. And these nanometer-sized man-made crystals all have unique optical properties, which are especially useful for bioimaging purposes. So these optical properties may include fluorescent spectra in both the visible and infrared range. So the skin has its own fluorescence, which essentially means when we shine light at a certain wavelength, it kind of glows. Like if you notice, like when you put a laser against your finger, or just like, you know, like a red laser pointer, that you can kind of see red through the finger. So there's like, the skin has its own interference. So when we're trying to do fluorescence imaging, whatever device we're using, ideally has the least amount of interference from your skin. So we focused on using quantum dots in the visible and infrared ranges. You can see on this chart as there's the least amount of interference. And there's even less interference in the near infrared range, which means that the imaging can penetrate deeper into your skin versus visible, which can probably only penetrate a small amount. So my goal was to analyze if there would be any change in the fluorescence spectra of the various quantum dots that we've been synthesizing with pH change in the range at which cancer differs from healthy cells. And so if this relationship can be found, then quantum dots can potentially be used as a method to detect cancer much earlier than many other therapies. So the first quantum dot that I used was reduced graphene oxide quantum dots, which I synthesized myself from RGO. The second was synthesized by Matt Daddett from glucose. The third was also synthesized by Matt Daddett by L-glutamic acid. And the last was synthesized by Dr. Roberto Gonzalez Rodriguez from glucose and urea, and is called aluminum doped reduced graphene oxide quantum dots. So once again, why these four? The reason is that they all fluoresce very well in the infrared range, and they all happen to also have peaks in the visible range. So in order to collect my data using a pH meter, I would take the pH of the sample of a 20 milliliter sample. I would do imaging with that sample, and then I would add acid or base in order to get intervals of 9 intervals between the pH of 6 and 8 for each sample. Once I was at the desired pH, I would put it into the imaging apparatus, and we had a few different imaging apparatuses we used. The first was the Symphony 2 spectral array for the visible range, and then just based on various setups, we had an 808 nanometer laser, a 400 nanometer, and 400 nanometer laser for the infrared range. And then we all charted them. So the results for the reduced graphene oxide quantum dots, they all had consistent spectra, which is good for other imaging purposes, but unfortunately their relative to the pH, there wasn't any particular pattern. I color coded from more acidic being red, then going Roy G. Biv. So the most basic is purple. As you can see, we kind of have purple on top, but then you have purple also kind of in the middle, and then you have yellow on the bottom, the visible, and you also have green on top, purple on the bottom, red somewhere in the middle for the infrared. Glucose graphene oxide quantum dots. Once again, they didn't have much of a pattern in the infrared spectrum, but in the visible spectrum, they had a notable pattern in that the intensity of the spectra increased with acidity. So the more acidic it got, the more intense the spectra became. L-glutamic acid was very similar in that the infrared spectrum was, once again, no definitive pattern, but the visible spectrum very clearly went from a gradient of more acidic being less intense and more basic being more intense. So kind of the opposite to glucose. And finally, the aluminum dope produced graphene oxide quantum dots, quite a mouthful, showed the most promise in that both the visible and infrared had notable patterns between pH and intensity, with the infrared showing a positive correlation with intensity and basicity, and the visible spectrum doing the opposite with increased intensity with increased acidity. And so this inverse relationship is potentially even more useful, as while intensity is relative for consistent conditions, even just using a different laser or maybe using a different area, intensity can be not necessarily like a set number for a certain molecule. So if you have multiple measurements that you know that there's a relationship between, such as visible and infrared, then it would be much more reliable to say that yes, this area is more acidic than another area. So our conclusions, there doesn't seem to be any pH correlation between RGQDs and their fluorescence spectra. There is notable visible pH sensitivity for GGQDs and LGGQDs, which merits further investigation. And infrared and visible pH sensitivity were both positive for aluminum RGQDs, which should definitely be further investigated. So our next steps are, as Matt previously stated, we need to do cytotoxicity studies for the GGQDs and LGGQDs, as if they show themselves to be toxic to biological environments, especially healthy ones, that could prove to be difficult to justify their use, as we're using them for detection, not for any type of target. We also would then, and we already performed those tests for RGQDs and AL RGQDs, and both of them have shown very low levels of toxicity. And then we would move on to live cell imaging studies. And so we would take a line of cancerous and non-cancerous cells, and then we would, recording their pH, add the various quantum dots and measure their fluorescence based on the pH and see if there was still a good correlation when they're in biological environments. Here are my references, and I would also at this time like to thank the College of Science and Engineering, Dr. Anton Namov, Matt Daddit, and the rest of my lab.
Thank you. We might have time for one question. Is there any questions from anyone? Okay. Okay, Veronica, well, thank you so much for your presentation. And I wanted to remind everyone that there is an award ceremony at 4 o'clock. So you can find that link at the SRS website. Thanks to all our presenters today. We appreciate your hard work and taking the time to share with us.